So in assignment 1 in exercise 3, we would be viewing some different microscope slides. Instead of doing that, what I thought might be more interesting for you would be to simulate an experiment where you took a scraping from between your teeth and gums with a toothpick and made a slide and did what we call a negative stain to stain the background of the slide to bring out all of the little details of the different types of bacteria that might be present on the microscope slide. And this is an image of exactly that sort of sample. So first thing I want you to notice is there's a whole lot of different things living in the plaque. In my previous video, I talked about some of the different shapes of bacteria. So here we definitely, next to this letter G, we've got something that is definitely a spirillum. And we have a whole bunch of those all over. They're kind of faint and they show up best with a negative stain. So that's kind of why we would have used this type of procedure. I also see some short bacillus shaped rods. I see some that might be a single coccus bacteria, but it could also be a bacillus just turned on its end. I see some here that look a little bit curved. So those could be a Vibrio shape, but it's difficult to tell because it's kind of out of focus. And then we have lots of these bacteria that have the pointy ends on them. Those are what you'd call fusiform bacteria. And in fact, tooth plaque is the main place that most people think of when they think of where do fusiform bacteria live. So now this is magnified under an oil immersion lens, so it's probably about 1000x total magnification. And then we've also taken that image that would be not that big, and we've zoomed in a lot so that you could see all this detail. The next slide that we would make would be to take a cotton swab and swab on the inside of your cheek and then make a slide out of that. And we would just do a normal stain on that because the cheek cells would stain pretty well. And this is also going to be under the microscope, although we would probably only be at 400x total magnification rather than 1000 because I'm looking at how small these bacteria look, and they're not really in focus either. So you're not going to see bacteria too well without the oil immersion lens. These large pink blobs with the dark centers in them, those are cells from the inside of someone's cheek. And the reason that you see multiple dark spots in the image is that the dark spots represent the nucleus in those cells. And if you see more than one spot on what looks like a structure, that cell might just be getting ready to divide. But I wanted you mostly to take away from this. Look how small these bacteria are compared to the cheek cell. Now, bacteria are prokaryotes, and human cells would be eukaryotic, meaning that they have a nucleus. And I just wanted you to take away the massive size difference between a prokaryotic cell and a eukaryotic cell. This whole thing is probably one eukaryotic cell. All of these tiny little speckles in this area here and on some other spots all over the image, these tiny little speckles, those are individual prokaryotic bacteria like we blew up in this slide. So that's just the difference between 1000x and 400x in seeing them. But I mean, This is just so big by comparison. And there's a lot of variation in how large a cell can be. We've talked about variations in shape, but cells can be from, um, 5 micrometers to on the order of a nanometer, all the way up to, um, oh, geez, if those are 5 micrometers, these look at least 1,000 times bigger than that. Even, in fact, even the nucleus of a eukaryotic cell is quite a bit larger than any of the prokaryotic cells that are on this slide.